Hello everyone, this is Ghairatullah Shinwari. Welcome to Medix YouTube channel. In today's video, I'll be talking about apoptosis. We have other videos about other medical lectures such as physiology and pathology. You can watch that on our YouTube channel. So let's continue about apoptosis. As I have mentioned, apoptosis. So what is apoptosis? Apoptosis is a genetically programmed cell death. So genetically programmed cell death. The second thing about it could occur to single cell, which means it could be a cell death of single cell or small group of cells. The next thing about it is that it could be physiological cell death or it could be a pathological cell death. Or pathologic. And the other thing which is very important and always remember about apoptosis is there is no inflammation after apoptosis. These are few things to begin with about apoptosis, but I will go to more details in this video about apoptosis. The mechanism which is used here in apoptosis, the difference between apoptosis and necrosis, why does it occur and how does it occur, the physiological reasons of apoptosis, the pathological reasons of apoptosis. So as I have mentioned few things to begin with here about apoptosis. As I mentioned, it's a genetically programmed cell death. And why would it occur? If you see in our body, there are millions and billions of cells, and each of these cells contribute to the function or the duty which our body provides. Let's say smelling or seeing something or thinking about something, which means focusing about something, or like movements, different movements that our body does. There are different things that I can talk about on and on about our body, the functions of our body. All these functions are actually carried out by our cells, which means if you have followed basic physiology lectures, so you would definitely know that the cell is basic structural and functional unit of our body. So why would the cell die? It could be a different reason. Could be different reasons behind the death of the cell. It could be a physiological reason, as I've mentioned, or pathological reason. In apoptosis, it's mostly physiological reason when a cell dies. So let's draw a small group of cells for better understanding. Let's pretend that this is a small group of cells and few of these cells needs to die. As I mentioned, reason could be physiological death or pathological death. But why would a cell die? There's a simple answer to it. A cell, if it could maintain its normal function and structure, it will never die unless there is a stimulation, a pathological stimulation or different kinds of stimulation, trauma, viruses, bacterial infections, or autoimmune diseases. But in normal physiological situation, a cell will not die unless there is a stimulation, as I've mentioned. So if one of these cells or the complete group of cells could not carry out their normal duty anymore, and it could not preserve their structure anymore, it will die. And in most physiological ways, the cell death which will occur, it will be apoptosis. So I will magnify one of these cells for better understanding. I have magnified one of these cells for better understanding. Let's pretend that this cell cannot preserve its structure, the anatomical structure which a cell has. Or, as we all know, there are different organelles inside of a cell, of which one is very important, called mitochondria. All of these organelles are actually very important for survival and functional purposes. But mitochondria is the most important in apoptosis, and the reason of it is that it is the stimulant for apoptosis, and the reason of apoptosis actually. But in intrinsic mitochondrial pathway, actually there are two pathways of apoptosis, which mean there are two ways by which apoptosis occur. The first way or pathway is called intrinsic mitochondrial pathway. So first pathway is intrinsic mitochondrial pathway. So how would a cell die of apoptosis? By intrinsic mitochondrial pathway. As I have mentioned, a cell which, if it could not do its function anymore, physiological function anymore, or it could not preserve the structure, anatomical structure of itself anymore, it will die. So let's say that this cell which I have drawn here, there is a damage to the cell membrane or to one of other organelles, like maybe a ribosome or a lysosome, which is another enzyme, and there is a damage to it, and damage to different parts of the cells, and the damage is so severe that the cell cannot recover itself from the damage. So the cell will eventually die, and when it dies of apoptosis, mostly this intrinsic mitochondria is responsible for it most of the time. Most of the time, this is the pathway. There is maybe, as I have said, cellular destruction to the cell membrane destruction, or to the organelles. The destruction is to the organelles or any other proteins available inside of the cells, and it needs to die eventually. And the way that it will die by intrinsic mitochondrial pathway, mainly the main responsible organelle would be mitochondria, which I have drawn here. So let's say this is our mitochondria. What is important about mitochondria, that there is an enzyme called cytochrome C. Cytochrome, it's not actually a single enzyme. There are different enzymes inside mitochondria, but the important among them is that cytochrome C. If cytochrome C is inside the mitochondria, and mitochondria would have two layers protecting the enzyme inside, keeping the enzyme inside, but the cell, if the cell, there is destruction to the cell, damage to the cell, irreversible damage to the cell, or as I have mentioned, the cell has lived out its usefulness and it cannot carry out the physiological function anymore. So what happens here in intrinsic mitochondrial pathway, I will remove other organelles for better understanding. There are there is a question: what's keeping the cytochrome C inside the mitochondria? There is a protein responsible for it, which is called BCL2. There are actually 20 different types of proteins available and responsible for different function inside the mitochondrial internal. Actually, to mention that there are two membranes of mitochondria: intrinsic membrane, internal membrane, and outer membrane. So BCL2, which is a protein, it's available in the internal membrane of mitochondria, and also BXCL, and there are different other proteins. So this BCL2 is available in internal mitochondrial membrane, and it has the job of keeping cytochrome C inside. But what happened in a cell which needs to die of apoptosis, which means the cell is old or there is irreversible damage to it, this BCL2 initially there will be a deficiency of it. The cell will become deficient of BCL2. So the cell will become deficient of BCL2, and also there is another enzyme called Buck and Bucks. Buck and Bucks, which is responsible, which will allow actually cytochrome C to leak outside of the mitochondria. So there is one protein or enzyme which is called BCL2, and then there is another BCL2 and BCLX, and then there is another pair which is called Buck and Bucks. One was responsible for keeping cytochrome C inside, while the other one is responsible for leaking or letting cytochrome C to come out of mitochondria. So how does with this Buck and Bucks allow cytochrome C to come outside? In the deficiency of BCL2, this Buck and Bucks will make actually a channel. Like if this is let's pretend this is a mitochondria, and there is, I will not draw the internal membrane just for better understanding. So this is cytochrome C. This Buck and Bucks actually will create channels, this kind of channels between outer membrane and inner membrane, and through these channel, these cytochrome C will leak out. So once they leak out, I will come back to the normal cell structure, actually this picture. Once these cytochrome C leaks out, what it will do now is that there are other proteins that this cytochrome C will active them, which are called caspases. So cytochrome C will initially first actually turn on APF1, which is a deactivated form of caspases, and APF1 will actually activate caspases. Which caspases? In intrinsic mitochondrial pathway, because caspases, it's just not a single protein, it's different proteins, it's like a combination of proteins. In intrinsic mitochondrial, the caspases which is activated is caspases 9, and through caspases 9 then the procedure continues. Caspases 9 will actually activate caspase 8, and 8 will actually activate 6, and 6 will activate 3. So what these caspases do is important, because these caspases will actually destroy anything which is inside the cell. So any cell component which is available inside will be destroyed by these proteins called caspases. And inside the cell, as we all know, there are different organelles, there are different proteins, there are it's like the cell membrane, the structure. These will be all destroyed by caspases. The other important thing about intrinsic mitochondrial apoptosis actually is that when the destruction of the cell is done here, and also that's the also reason for no inflammation. That's why there is no inflammation after apoptosis. That's the main reason. When this destruction of the cell is done through apoptosis, apoptotic bodies are generated or created. This is actually the organelles inside the cell and also the cell itself. These apoptotic bodies, which are like, let's say, organelles, parts of organelles, proteins, enzymes, whatever is inside the cell, plus the membrane of the cell, these all apoptotic bodies are membrane bound. which means there is a bilayer phospholipid membrane and it has bounded these organelles and proteins, and that's the reason behind apoptosis causing no inflammation. Because if your immune system see anything which has to be inside of the cell, like protein, DNA, whatever it is, and our body sees it's outside of the cell and it has no membrane attached to it, what our body does is that it will show an immune response, and that immune response is called inflammation. So that's why there is no inflammation after apoptosis. That's the main reason. So once these apoptotic bodies are generated, created, the next thing which happens is that these apoptotic bodies, which are now in our internal system, and it has died and there is no usefulness to them anymore, it needs to be cleaned. So who is responsible for cleaning these apoptotic bodies? Normally phagocytes, like macrophage. So they will clean these apoptotic bodies, and new generated cells or other cells which are new and they can carry out the physiological function, they will come and replace these dead cells. So this was our intrinsic mitochondrial pathway of apoptosis. Next, I'm going to be talking about extrinsic mitochondrial pathway of apoptosis. For that purpose, I need to draw another cell. So next, I'll be talking about extrinsic mitochondrial pathway. Extrinsic mitochondrial pathway. Mitochondrial pathway. What happens here is, as you have seen in intrinsic mitochondrial pathway, the stimulation of cell death was from inside the cell itself, like there was cytochrome C inside and it leaked out to the cytoplasm, activated caspases, and the cell death occur. But in extrinsic mitochondrial pathway, the stimulation for the cell death apoptosis comes from outside. So let's pretend that the cell it's not useful for our body anymore, and it needs to die, and the reason could be physiological or pathologic. But this pathway, extrinsic mitochondrial pathway, is also called Fuz ligand and Fuz receptor pathway. There are receptors on cell surfaces. Let's say this is a receptor called Fuz receptor, and there are ligands on other cells such as T lymphocytes called Fuz receptor. Fuz receptor. Fuz actually ligand. So Fuz receptor was on a cell which needs to die, and Fuz ligand was on a cell which will actually stimulate the cell death of the particular cell which needs to die. So let's say this is a T lymphocyte and this is a cell, target cell, which needs to die. What happens in extrinsic mitochondrial pathway, this Fuz ligand binds to Fuz receptor, activating caspases inside of the cell. And the caspases which is activated through Fuz ligand, Fuz receptor pathway or extrinsic mitochondrial pathway is caspases 8. And then initially it will go through the same procedure. 6 is activated, 3 is activated after that, and all this caspase cascade is activated, and then the same thing happen. Destruction of the cellular component, the the organelles which is inside the cell, proteins, enzymes, whatever is inside the cell, and eventually apoptotic bodies are created which are membrane bounded. And these apoptotic bodies are again cleared by macrophages. So macrophages again will clear these apoptotic bodies. So there is a question behind it. What happens that the macrophages normally in our in apoptosis there is no immune response, but once these cells are dead and apoptotic bodies are formed, macrophages will come to the area. So what causes the invitation of macrophages to the area? Let's talk about it. On a normal cell, as I have mentioned, normally cells would have bilayer. So if I draw another layer, it will be inside of the cell. Actually, cell structure or the anatomy of the cell is not as I have drawn it, but it's just for better understanding. So as I've mentioned, what happens, what's the invitation for macrophages to come to this area and actually eat or phagocytose this cell which has died and apoptotic bodies? There are different reasons. The first thing is that the cell which is dying, it will release proteins from its membrane. Second thing about it is that the inner cell membrane, there are proteins available. It's actually not a protein, it's a phospholipid called phosphatidylserine. Phosphatidylserine. In a normal cell, this phosphatidylserine phospholipid is available in the internal cellular membrane. It's available here. But once the cell is damaged beyond repair, the phosphatidylserine phospholipid will be expressed on the outer membrane. And the reason of this is the cellular destruction actually, or abnormal cell structure, or the cell is old and it cannot carry out the physiological functions anymore. So once these two steps happens, these are the invitation, both of them are the invitation for macrophage. Invitation for max actually. So macrophage will come and phagocytose this particular cell or group of cells, and it will clear like nothing happened, which means there will be no inflammation after this cell death. And this is the reason that there is no inflammation after apoptosis. So now I'll be talking about difference between necrosis and apoptosis. On one side I have apoptosis, on the other side I have necrosis. So apoptosis is genetically programmed. Genetically programmed. And necrosis, it's not genetically programmed. Apoptosis could be physiologic or pathologic. While necrosis is always pathologic. No inflammation after apoptosis, always inflammation. single cell or small group affected by apoptosis, and in necrosis, large group of cell. So these are the major differences between apoptosis and necrosis, and as I have mentioned in my video that there are few differences between apoptosis and necrosis. Now I'll talk about the physiological stimulants of necrosis and the pathological stimulants of apoptosis. So now let's talk about physiologic and pathologic stimulants of apoptosis. In physiologic apoptosis, there are different examples which one can use, like during embryogenesis. The procedure which cell dies and new cell replaces the dead cells is apoptosis during embryogenesis. So the cell death basically during embryogenesis is apoptosis. Endometrial shedding. Endometrial shedding during menstrual cycle is also a cell death by apoptosis. I will draw a picture of endometrium here for better understanding. So if this is a uterus, during menstrual cycle, the endometrium, which is, if I go in details, the uterus has 3 layers actually: endometrium, myometrium and perimetrium. During menstrual cycle, the bleeding which occurs, the endometrium, this part of, let's say this is my external layer, so the cells which are available here, they die and they shed with menstrual bleeding. So the way that these endometrial cells die is called apoptosis. So this is actually a physiologic apoptosis. And another example which I can give is RBC. After each 120 days, our RBC is renewed. Old RBC die and there are other RBC created, produced in the bone marrow, and it will replace these old ones. This is also actually a physiological apoptosis. So now I'll go to pathological stimulants of apoptosis. There are more physiological stimulants of apoptosis, but as I have just mentioned few of them here. For more details and information about this video, you can also go and study and have a look at the Harsh Mohan or Robbins pathology, Patoma or BRS. So about pathological apoptosis, I'll give a few examples as well. So in pathological apoptosis, there are different examples which I can provide, but I will go through a few of them here. In CNS, disease called Parkinson's disease. While this disease occurs, the cell death during this disease is through apoptosis. Alzheimer, another disease in CNS, same thing, apoptosis. Dementia, in dementia the cell death is also apoptosis. And also severe DNA damage also triggers apoptosis. These are few examples of pathological apoptosis. And also a very important thing about apoptosis is tumors, cancer. So there is also a question in everyone's mind that, so if the cell cannot do its function properly, and there is a difference, or there is change in the structure of the cell, apoptosis occurs. But why does apoptosis do not occur in tumors, in cancer? Cancer has a special thing in it, which is that they blocks, cancer blocks apoptosis through bringing down BCL, actually bringing up, increasing BCL2 on their surface, internal mitochondrial membrane, actually not surface. Cells which are tumor, like which are cancer cells, they will increase BCL2, which is available in the internal mitochondrial membrane, and BCL2 will not allow cytochrome C to leak to cytoplasm and activate caspases. So they will deny apoptosis through increasing BCL2 and decreasing BAC and BUX, which were responsible for transport of cytochrome C. So this was apoptosis in detail. For more medical videos, please like and subscribe our YouTube channel and like our Facebook page.